Hi everyone, welcome back. Carrying on with some organisation today, looking at enzymes part 2, denaturing and rates of reaction. So what we're going to be having a look at today is what denaturing is, what can cause an enzyme to denature, and how you can make an enzyme controlled reaction go faster or slower, mainly looking at the effect of temperature and pH, which is what you'd be expected to look at in practical exams as well. So as always, grab some paper, grab those pens and follow along with me. So before we get on to looking at new stuff in denaturing, I think it's important we go back and look at the structure of enzymes and the lock and key hypothesis first, because those are really important to understanding what denature is. So if you've sort of forgotten what happens in the lock and key theory, I go back to the previous video, I'll link it in the description and have a look at that first. So you've got the main structure of the enzyme, which is the bit in green. You've got the substrate next to it. If we were talking about digestion, substrate would be like a big bit of protein that you're going to break down. And then in your enzyme, you've got the part called the active site where the substrate fits. Last lesson, we also talked about the fact that enzymes and substrate fit together exactly like a lock and a key. So the active site is a different shape depending on what that enzyme works on. So for example, if you were protease, you would have a specific shape that would only work on protein. If you were lipase, you'd have a shape that only work on lipids. So out of these two ones, the top one isn't going to fit because it is the wrong shape for that enzyme, and the one on the bottom will fit because you've got the sort of because you've got the triangle on top, the half circle on the bottom, so it's the right shape. And having the correct shape has a special term, we call this specific. And every single enzyme will have a specific shape depending on what it works on. So all the ones in your digestive system are specific for the protein or carb or lipid that they break down. So as well as having a specific shape, enzymes are very, very fussy little balls of protein. So if we think of a temperature scale here, I'm going to put too cold on one side and too hot on the other. These are thermometers I'm drawing, I promise. Now somewhere on this temperature line, the enzyme's going to have a temperature that it works at best. So this temperature in the middle is our optimum, the temperature the enzyme works best at. Okay, so let's look at a scenario where the enzyme is in condition that is too cold, or you've started a reaction off at a low temperature and then you're steadily increasing it. So at low temperature, particles are still moving, however they have much lower store of kinetic energy. So they're moving slower, and if they do collide, it's more like bumping into someone in the corridor as opposed to, ramming into someone with a car. You're going to knock into each other, but not much is going to happen. So in terms of particles and in a chemical reaction, if particles do hit together, that's great, but unless they're hitting together with enough energy, those aren't going to react. So if particles come into contact and they don't have much energy, they're not going to bind, that's going to be a slower reaction. If we were to look at this on a graph, or imagine that as the line goes up, the reaction's happening faster, you'd see it's a gradual increase. As you gradually increase the temperature, you're adding more kinetic energy to those particles until you reach this sweet spot at the top, the peak of the reaction, and again we call this the optimum. So this is where you've got the perfect balance between particles moving fast and then bumping in together in a meaningful way for them to react. Imagine you are smashing cars together this time as opposed to bumping someone in the corridor. So these particles are going to have enough energy to then react. This would be a scenario where the substrate and the enzyme fit together perfectly. They've got enough energy to do so, sort of lock into place like a couple of Legos, and then the enzyme will do its thing. Once we get past this optimum though, you'll often notice that the rate of the reaction suddenly starts to dip, and we've got to work out why. Why does this happen? So look at this, So let's look at the scenario where our enzymes are put into conditions that are too hot. If you make an enzyme too hot, it will be destroyed. It has a special scientific term called denatured, but this just really means that the enzyme shape has changed in such a way that it won't be able to fit the substrate in. It's like, you know, getting a Lego piece that's been a bit melted, it's never going to fit in the same way again. So if we take our general model of an enzyme, it looks a bit like a Pac-Man or a little mouth, and we sort of look at it in a bit more detail, rather than being a sort of C shape, your enzymes are just a big tangled ball of protein. So I'm going to draw them as like a little sphere, little interconnected dots. Those dots will be our amino acids, and the lines will be the bonds, so the forces that hold those bits of protein together. And you can see the active site on each here in orange. So what we're going to do now is we're going to put our little enzyme into a saucepan. This is not a real enzyme. Enzymes aren't that big. But I'm going to imagine we're going to put this happy little enzyme and we're going to turn up the heat. We're going to boil this little enzyme alive. So what heating is going to do is it's going to add more energy, make those particles move faster, so the particles inside the protein are going to vibrate more, and then something bad's going to happen. So what's happened here is some of those bonds have been broken by heating the substance the enzyme is in. The particles are moving so fast that they actually break apart. So therefore, the shape of the enzyme's been destroyed, and the active site's going to change shape, so that enzyme will no longer fit the substrate, and that reaction will not be sped up. Okay, so if we look at our graph again then, the reason why that peak is going down so fast is because of this shape change. The enzyme's been denatured. It cannot speed that reaction up anymore. So if we look at this in basically a graph form, you might see it in an exam, you'll see that the rate is going to steadily increase, and then you're going to have an optimum, this peak, and then it's going to steadily drop. It often looks like a bell-shaped curve. And remember, different enzymes are going to have different temperatures at which they have their optimum conditions. Though ones in our body, like our digestive enzymes, are going to be around body temperature 36 to 37 degrees C. Enzymes are also really fussy about the pH that they're put into, not just temperature. And remember, the pH is just how acid or alkali conditions are. So we're going to look at a pH scale this time, remembering that we go from 1 to 14, 1 being strong acids and 14 being strong alkali, and 7 is in the middle, that is neutral. So we looked at some conditions in our digestive system in a previous lesson. In the stomach you've got acid, so that's going to be a low pH around 2. Your saliva, your spit is around pH 7, neutral, and the conditions in your small intestine are alkaline. And each of these areas will have a different enzyme that works there. So protease will have its optimum pH around 2 due to the acidic conditions. Your saliva, the amylase in there will have an optimum of around 7, and in your small intestine, your carbohydrates will have a pH that is the same as the conditions of the small intestine. So what happens when we take an enzyme and put it in the wrong pH? So we're going to imagine we're going to take a protease enzyme, one that you find in your stomach that likes acidic conditions. We've got its active site here in orange again, and it's in a good mood today. What we're going to do then is take this protease enzyme and put it in the wrong pH. We're going to put it in pH 5. And oh god, look, what's happened is the bonds have been broken again. So something about this conditions has caused those bonds to break, and now this enzyme isn't going to work anymore. So this enzyme has been denatured. Okay, so let's have one last look at these graphs then. I know graphs aren't that fun, but they do come up a lot in exams. So we're going to have temperature and pH on the bottom on our x-axis and rate on our y. Temperature has this, again, bell-shaped curve. It goes up and down. Sometimes the rate falls quicker after the peak. The steeper the line, the faster the rate is falling. And if you have pH, you'll often have smaller peaks or separate peaks. This is due to enzymes having very different pHs at which they work at best. So if you look at this one and you have to guess which one was the enzyme that's most likely to be protease and most likely to be carbohydrates, hopefully you guessed that X is going to be protease because that's found in the stomach, so it's acidic, and carbohydrates, that works in the small intestine, so the conditions there are going to be alkaline. Thanks so much for watching. Hopefully that you got something out of this video. Make sure that you get some notes down on the different types of enzymes, how they're denatured, what denaturing is, and what happens when you change pH and change temperature. Make sure you like and subscribe for some more as well. It does help, and I will see you all very soon.